Well, good morning, good afternoon, good evening, everyone, and welcome to the world's most exciting classroom. My name is Joe Cutler, and I'll be your host for today. And this is the first time I have said that in 2025. So, right before the Christmas break, we took a little pause while the ship was in Christchurch to celebrate the Christmas holidays. Obviously, classrooms were out of school, but now we are back in action, and we're going to continue with our regular events every Thursday, right up until July when the ship returns to Falmouth in the UK. So I can't believe that all the way back in August 2023, we left Plymouth in the United Kingdom. We've sailed across the Atlantic, all around South America, across the Pacific Ocean. We've been hanging out in New Zealand and Australia. And now the Oosterschelde and the Darwin 200 team are making their way back across the Pacific Ocean to the Falklands. So let's take a little look at our map and let's see what's going on here. So let me make that a little bit bigger. There we go. You can see that right before the Christmas break, we were hanging out in Christchurch. The ship was there over Christmas. They celebrated Christmas there, New Year's as well. And then on January 2nd, it set sail. So I've got a few little pictures here that we can grab right from the map. So if you visit darwin200.com, you can always visit the map and you can see what we've been up to. The latest pictures, the latest video clips are uploaded there. You can see the ship leaving Christchurch here. Here's a few of the passengers who are on board right now. Now, the plan was it was going to be a few days of smooth sailing to get ready for the bigger journey so that people could learn how to work the sails and the ropes and things like that. Because this is going to be a 7-week journey across some of the roughest waters on the planet. If I back out a little bit here, you can see what I'm talking about. We're going to go all the way around here through the roaring 40s. We're going to round the horn, and that's our destination in the Falklands. But Mother Nature doesn't always cooperate, and immediately they were in some pretty rough weather. Here's a little video clip. So as you can imagine, that's a pretty tough adjustment to a brand new crew just getting on board. There was a lot of seasickness going around. But at the same time, it was a pretty good team building thing to have to get right into it and right into that kind of weather, which really is kind of a precursor for what's probably coming in the next few weeks. If we continue on, you can see that weather didn't last, and actually they lost the wind. So they had to take this massive detour, this bump here to the north. And we're going to have a little video connection from the ship. And they will tell us exactly why they had to do that. But if we look at a couple more things, let's see. You can see the weather calmed down, which would have been nice for them. A nice sunset there on the Pacific. You can see that there they are working some of the ropes. So they're getting the hang of what it takes to pilot the ship, bring different masts up and down depending on what the wind is doing. They've got laundry to do on board. Oh, that's not the right clip, but this is one where they're just doing repair on one of the sails. And then one of these is a clip of, there we go. So being on board for 7 weeks means you can't bring enough clothes for the entire time. So they have their washing days. They've got to get out their buckets and then hang their clothes all over the ship to dry them out. So there is no laundry facilities. They've got to do it by hand out there on the ocean. So I did mention that I have a little clip from Tom and Mara, who's our researcher on board right now. So let me stop that screen share and let's get that little clip up on the screen and let's see the update we have from the ship. So just bear with me for one moment. There it is. And here we go. Well, it's been a little bit since we've caught up with the Oosterschelde. They have left Christchurch after spending the holidays there and are now making their way across the Pacific towards the Falklands. They still have a long way to go, as you can see from the map we looked at earlier. But we are going to make a nice little connection with them now. We've got Tom and Mara. We haven't met Mara yet, so we're going to get a chance to meet Mara. So let's go on deck right now. Hey, Tom. Hey, Mara. How are you?
Hello, how are you doing? We're all good.
Yeah, good. Good. It's great to see the two of you. It looks like a beautiful day today.
Yeah, it's a beautiful sunny day. We haven't got much wind today, so we're motoring but it's a good day to do some laundry.
All right. Well, Tom, you're a very familiar face. We've had you join us tons of time, but this is our first chance to connect with Mara. So Mara, it'd be awesome if you took a minute and introduce yourself.
Yeah, sure. So I'm Mara. I'm a marine biologist, currently doing my PhD at the University of Worcester in the UK. And I joined this expedition very last minute. So I was already in New Zealand doing some work there for my PhD. And then this opportunity came up, and I found out 2 weeks before we left Christchurch that I was going to be joining. So yeah, super excited to be on board and to get stuck into the project.
Yeah, that's great. And so you're, part of your duties on board will be continue the bird survey that we've been undergoing all the way across the planet. How's that been going so far? Are you getting much action?
Yeah, it's been really exciting actually, getting stuck into that. So we've been doing about an hour each morning and each evening, and we've had some really cool species already. As soon as we left New Zealand, there was a lot of action. We've had a lot of big birds like albatrosses. We've got a lot of different species of petrels, and quite a few that have come up as quite rare as well.
Yeah.
So it's been really cool to see what's around here.
Awesome. Good stuff. And then, Mara, I always like to ask this question. How were the first few days? I know it takes some people a little bit to get their sea legs. Did you take to it pretty fast?
So the sea didn't really ease us into it. As soon as we left New Zealand, it got pretty rough pretty quickly. So it took a few days to get used to it, but I luckily didn't have any issues with seasickness. So I felt very grateful for that. But it took a few days to get my sea legs, and after a few days it eased off a little bit as well, which was nice.
Yeah.
But definitely got onto the swing of things now.
Awesome. And Tom, last time we caught up, you were in Tassie. You were climbing trees. How did that go?
That was very, very cool. So we're doing a project about the old growth forests of Tasmania, and as part of that, we climbed to the top of one of the 60-meter eucalyptus globulus, which are these towering trees that shoot up right above the canopy. We went up there. There's a team of 3 of us. 4. And we were there measuring doing wraps of the tree, getting measurements of that, so we can calculate the volume of the tree, which they use to classify them as giants and protect them. So that was pretty cool to experience all of that.
Yeah, how was the climbing? Was it about what you expected, or was it pretty tough going?
It was pretty tough, to be fair. It was a long way up. It's all right for a few meters, but then you realize when you look up, you're like, there's a long way still to go. So definitely interesting for sure.
Yeah, very cool. I'm going to bring the map up for a second because I want to talk a little bit about the journey since Christchurch. Before we do, can we get a little pan out? Can we see some ocean action?
Sure. Here we are.
Beautiful. Any marine life since you left Christchurch?
We've had a little bit. We had some dolphins yesterday. Just some short-beaked common dolphins. They came and played in the bowway for a little bit. That's I think the first dolphins we saw on the trip.
Yeah, this trip.
This trip. So everyone got really excited, and most people were up on the deck anyway, so it's nice. Most people got to see them. And then we've had 2 whales.
2 groups of whales.
2 groups of whales. Yeah.
Different days.
Had some sei whales.
Great.
But that's it so far, I think.
Yeah, very cool. I'm going to pop the map up here. We're going to look at the map. Tom, I know you had a break after Tassie. Were you home for the holidays, or did you spend holidays on the ship in Christchurch?
I met up with my family abroad, which was nice, for Christmas. So that was great, up in Thailand actually. So that's pretty special. Nice. But then came back to the ship in New Zealand for New Year's. So back with the crew on board.
All right, very cool. Well, the first thing I want to ask about, because it looks like you start off in the right direction, then there's that big hump in the middle. What happened?
Yeah, well, being such a long voyage, and we were trying to sail as much as we can, so we obviously have to follow the weather systems. And the weather system that we got offered was this one that was taking us quite far north. So it was like we followed this big low pressure system around, hoping that it would take us more east. But yeah, we kept on going and followed it around, and now we're just crossing between low pressure and high pressure to catch the edge of another one.
Yeah. And do you get a sense from the captain, this is a big leg of the journey. Do you get a sense that you're on track, a little behind, ahead of schedule? How are things going?
I think we're pretty on schedule, yeah. They left 7 weeks for the entire journey, which is a long time. I mean, I think it was 5,200 miles that we have to cover, so it's also a big distance we have to go, but there's plenty of leeway in there for
and they factored in that we might have to go on some detours, and to do the official Cape Horn crossing, we also have to sail for 3,000 miles at least, going around it. So I think they factored in that before that we might not be able to sail the whole time, and would have to obviously go and follow the weather systems.
All right, very cool. Well, before we let you go, I want to know, these waters have a reputation for being some of the roughest on the planet. How have you found it so far? Is it living up to it, or do you think the worst is yet to come?
I mean, the first 2 days were definitely, I think people were maybe slightly worried that it's going to be like that for the next 7 weeks, because that was pretty rolly, pretty choppy. But I mean, at the moment we're meant to be in the roaring 40s, and it's very calm. So
people are likening it to the boring 40s.
Yeah.
Fair enough.
Yeah, I'm sure it'll change again. And, we'll see.
Yeah, I have a feeling we'll get some video of a little storm action.
Yeah.
I'm sure you will, yeah.
Everyone's enjoying the respite for a bit.
Yeah.
Yeah, very cool. All right, well, I will let the two of you get back to your day. It looks like with the sails down, it might be a bit of a relaxing one, but hey, sun, ocean, what could be better?
Cheers, thanks, Joe.
All right, guys, thanks so much for connecting. We'll let you go for now.
Thank you.
See you later.
Bye.
All right, so we had that connection with Tom and Mara last night, because right now it's night for them. They're on the other side of the world. However, every day they're getting an hour closer to our time zone, which means it won't be too long until we can have the ship back live with us in some of the world's most exciting classroom events. We don't have to rely on those recordings a couple days before. So pretty soon, as they continue to cross more of the ocean, each day they're almost getting an hour closer to us as they cross the different time zones. So we'll have them live with us within the next few weeks, which will be pretty cool. So as you know, we always love to have a guest speaker hang out with us and spend some time, share their adventures and what they do around the world. And we've got a great one to kick off the 2025 season. We have my friend Joe Cutler joining us. He is an ichthyologist. He is a National Geographic Explorer, and he has covered thousands and thousands of miles on rivers in Africa, in Gabon, in Zambia, Zimbabwe, Angola, doing biodiversity surveys. So exploring these incredible rivers out with teams, collecting fish, looking at biodiversity. It's pretty wild. And the last few years, through my organization, Exploring by the Seat of Your Pants, we've been able to host Joe on the banks of rivers from all over the world. So we're getting him in a bit of a different environment this time. We're getting him in a lab environment. So I'm going to bring in Joe live with us right now. Here we go. Hey, Joe, how are you?
Hi, Joe. How you doing?
Yeah, good. It's good to see you. It's been a while.
Yeah, thanks for connecting. I believe the last time we did a live stream, I was on the Kahoot River in Zambia. So it's good to reconnect, and I can continue telling the story of what's going on with the fishes we were collecting on the Kahoot.
Perfect. Well, Joe, let's get started. First of all, tell us where you are, and then let's get into that story.
Yeah, so right now I'm in the ichthyology collection at the American Museum of Natural History in New York City. So I'm in the ichthyology prep lab, and I'm just going to quickly slide out of the way. You can see there's a bunch of jars and equipment and more jars, and all of those jars are full of fishes here. And so I'm here organizing all the fish that my team and I have collected in Zambia over the last 2 years. It's about 5,500 fish. So all those jars are full of our fish, which is pretty darn exciting. And now that they're here in the Museum of Natural History, we can actually really finalize our identifications, figure out which of those fish are new to science, which are really interesting, and then we can study them in depth and describe new species. So I want to, for those of you who don't know about my background and the work that I've been doing, I'm going to share a few slides here, and then I will come back and we can look at some of the fish together before the end of the call and taking some questions. So let me just share my screen right now. And let me know, Joe, when you can see the whole screen.
Looks good, Joe.
All right, great. So I'm going to be talking a bit about the work that we've been doing exploring the wild Zambezi River. As Joe mentioned, my name is Joe also. I'm Joe Cutler. I'm an ichthyologist and freshwater explorer. I've worked mostly in sub-Saharan Africa, and over the last few years, really extensively in Southern Africa, Zambia, Zimbabwe, Angola, etc. And I am a total fish geek. So this slide shows some of the photographs of fishes that we collected on expedition. And one of the things that's really exciting to me as a scientist, as an explorer, is going into these really unique river systems, these remote, isolated areas that maybe have never been studied before, and being able to study the fish diversity. It's really exciting, and we are finding a lot of new species. So I currently work for the Wilderness Project. This is an NGO that specializes in setting river baselines. So we paddle down river systems, we study the health of the ecosystem, the diversity, the human impacts, and we use it to kind of evaluate the health and then come up with recommendations on how to protect those systems looking forward. So just a quick geographic reminder, the Zambezi River is one of the major river systems in Africa. It drains into the Indian Ocean in Mozambique, but it draws its source in these Angolan highlands and on the Zambia-Congo border, as you can see here. And our goal as a project is to study not just the main Zambezi, but its tributaries. So over the last couple of years, we have paddled everything that you see highlighted in red here. So we have taken the main Zambezi all the way from the source out to the ocean. We've taken the longest tributary, the Kahoot River, all the way down, and the Lungwe Vungu, the true source of the Zambezi. We've also surveyed that. And this year in 2025, we should be completing all the remaining rivers that you see here. So personally, I have paddled over 4,000 kilometers in the Zambezi Basin, and I'm really starting to understand the system. So this photograph shows some of my team members. We do these expeditions in canoes and kayaks or inflatables, depending on the water conditions. And we set out as far upstream as we can, and we paddle all the way until we reach the ocean. So these are really big adventures, lots of challenges, lots of exploration, lots of fun. And our goal is to study and understand the system. So we are looking at biodiversity, human health, but also things that you can't see with your naked eyes. So we're looking at water quality, environmental DNA, and heavy metal accumulation in these systems. And really, we want to know what's the state of the ecosystem. Are they threatened? What areas are particularly threatened, and what areas are pristine, so that we can come up with strategies to both protect the pristine areas and fix the areas that are degraded. And for me, one of the great things that we get to do is study fish on these rivers. So every moment I get, I'm either out with my cast net looking to catch a few fish, or out after dark with my dip net, also looking to catch a few fish. And there's a theme there. Usually I only get to do the fish work right at the end of the day, and it's after the sun's down, so you're dodging the crocodiles, but it's always worth it to get a bucket full of fish because, you know, in these areas in sub-Saharan Africa, there is an incredible amount of diversity in the freshwater fishes. And you never know what you're going to catch until you pull your nets and until you really get a good look at those fish. You really don't know what it is. It feels to me like we're on the cutting edge of exploration, science, and conservation. So it's a really cool place to be, a really cool place to work. And these fish that you see here in the photograph, we're taking photographs of the fish, we're taking DNA vouchers, and then the fish themselves are preserved and transported here to New York City, where they will live in perpetuity as kind of a resource library. So I just wanted to, highlight some of my team members. This crew you see here, we spent 84 days together paddling down the Kafue River. Almost all of us had capsizes. A few of us ended up in the hospital. It was a big, hard expedition, but we pulled through because of our teamwork and camaraderie, and I couldn't have done it without my team members. So we had some challenges. We struggled through a lot together. You know, when we were starting, the grasses were 10 feet high. Everything was soaking wet. When we finally got into the river was so choked that we're spending all of our time hacking our way through the brush with machetes and saws. We had to get through these barriers, both natural and man-made. So this is a big fish weir that was blocking the entirety of one of the rivers we surveyed last year. And we had to open a little hole in the fish weir and push our boats through. And then we encountered these incredible wetland areas. So, vast 100-kilometer, 400-kilometer wetlands where in some areas the river kind of disappears and you're just fighting your way through these wet grasslands full of leeches. So that was tough. And then lots of rapids. So we spent a lot of time looking at rapids, trying to figure out whether it's safe to paddle or not. In most scenarios, it's not safe. So we've got to unload our boats and carry our gear around. And that's exhausting. So there are times where you try to wiggle your boats down through the rocks of the rapids, and you just hope to keep everything afloat, because if you don't, it's a lot of equipment wet and a lot of discomfort and hardship. And you always have to be aware in the Zambezi Basin of Nile crocodiles. Now, Nile crocodiles are the second biggest crocodile species in the world, and they probably account for more crocodile-related deaths than any other species, and they are certainly keen to eat whatever's floating down the river. This photograph shows some hippos. These are our other competition in the river. So in the background there, all those gray things you see in the water, those are all hippopotamus. And sometimes they let you paddle by no problem, and sometimes they come running at you like a freight train. So always a little bit nerve-wracking. And this is just to show the size of some of the crocodiles. Crocodiles on the Zambezi get to be over 18 feet long, and at that point they are definitely man-eaters. So this is a hippo jawbone. We actually found a skeletized hippo on one of the rapids that we were surveying, and you can just imagine that you don't want to be bit by one of those guys. But we had other challenges. So this photo shows my hand right next to a lion track. So one of our nights in camp, everybody's sleeping soundly in their tents, and the lions are just circling around our tents all night. Thankfully, they didn't wake us up. And this is another track. This is actually an elephant track to give you some size, some scale on how big these animals are on the Zambezi system. Now, we were really lucky this year. We saw elephants, and we saw warthogs and lions and leopards and impala, all from the river. It was really incredible, really great wildlife viewing and exploration. But we also had challenges. Some of these areas are just full of mud. So you take a couple steps on dry land and next thing you know, you're knee deep. And I spent a lot of time with muddy feet this past year. But after having surveyed these river systems, I've got some major takeaways of threats to the ecosystems. So, we may think that these wild African rivers are, really pristine, really untouched by humanity, but in almost all these river systems, we're seeing evidence of overfishing, primarily through the use of monofilament gill netting. So these are large-scale commercial fisheries on small, isolated freshwater ecosystems, definitely unsustainable. We saw a lot of impacts of pollution, in some cases associated with mining. So this is a copper mine on the Kahoot River, and you can see how much it is degrading the water quality there. And this is trash pollution. So in the town of Kitwe, we noticed that there was a spike in plastic pollution, and the trees in that area looked like, kind of a Christmas tree covered in plastic waste. So that was really hard to see in such a previously healthy ecosystem. These systems are also heavily impacted by development. So this shows a hydropower dam, and this is important for regulating the rivers, for regulating flood pulses, and generating electricity, but it really dramatically changes the river ecosystem, and in particular, the floodplain systems on the Kahoot River. This is another hydropower dam. And you can imagine if you were a little fish living in the river, how much the introduction of a dam like this would change your ecosystem. One of the other impacts we're seeing on these ecosystems is invasive species. So this is a crayfish native to Australia, and we are unfortunately finding these throughout the Zambezi system, and we are monitoring to see if they extend into some of the other nearby basins. Finally, we're seeing a lot of evidence of new diseases. So there's a fish disease that's gotten into the Zambezi system called epizootic ulcerative syndrome. And it seems to be really affecting the cichlids and some of the catfish species, causing them to die in the environment. So that is some of the impacts and threats. And I think that's one of the reasons why it's so important what we're doing, why we are getting these baselines today, why we are studying the diversity today, because these rivers are rapidly changing. And if we don't study them now, they may change so quickly that we may miss those rapid changes in the system. And we want to be there in real time, studying, monitoring, and protecting these systems. So with that, I just want to say thank you, and I want to bring the camera back to me, and I want to show you some of the highlights of the fishes we've got here. So let me stop my screen share and come on back. You got me back here, Joe?
We got you back, Joe.
Great. All right. So I'm going to just reach behind me and I'm going to grab some fish, some highlights from the Kahoot River. So I've pulled out a few fish here. This is an African pike, a Hepsatus. This is one of the big predators in the Kahoot River system. It's also found in the Zambezi. But in the Kahoot, this is the top fish predator. And they've got really big teeth, a little hard to see, but they are just beautiful fish, very cool. This fish will have its own jar here in the museum. So that's a fun one. Let me show off a couple catfishes. So I really like catfish. This is very likely an undescribed species of catfish we collected last year, very close to the Kahoot source. You can see how elongate and thin this catfish is. It is a Claridae, very interesting fish. Nothing really like it known from the Kahoot or Zambezi system. So that's exciting. And a relatively big one I'm going to show you a relatively small catfish here now. So that is an adult Cheiloglanus catfish. So this is a tiny little catfish that lives in the rapids, and it's got a little sucker on its mouth, and so it can suck onto the rocks when the fast-flowing water is going over its body. And I imagine that there's a lot of undescribed diversity in the rapids-adapted catfish from the Zambezi. So very exciting to catch these specimens, and looking forward to finding some more as we get back into the field. Now I'm going to grab some minnows here, and I think this is a really good example of the importance of a collection like this at the American Museum. So I have 7 minnows here, and they may all look quite similar. Let me just start pulling them up. You know, this one's got a little line and a little spot on the tail. This one's got beautiful little striping pattern and a little post-humeral spot there. But, you know, they all look pretty similar, don't they? You know, they're all minnows, right? But when I look at these fish side by side, I actually can really start to distinguish some differences. You know, this fish is a little bit more elongate. This one's got kind of a hump in its back here, different shape on the fin. And so having fish side by side like this, having specimen that I can compare, that I can use a microscope to really look at what the differences are in these fish, allows me to distinguish species and allows me to identify which ones in this collection may actually be new to science. So excitingly, this little minnow, while it doesn't look like much, is actually an undescribed species as well. We know it, but no one has formally described it. So right now we just call it Anteromius purple stripe. It is a very beautiful fish while it's alive. When it's preserved, it looks just a lot like the other minnows. So I'm going to put these minnows away, but there's a lot of cool fish around the collection here. So right next to me, I've got a few other jars. So let me just sneak somebody else's fish out here. So this is a deep water fish here, and it was collected in Angola in 1927, if you can believe it. So these fish collections all around the world serve kind of as the library of life. So folks like me, ichthyologists, we go out and we make our collections. The collections come to a repository like this, they get curated, and then they get put into the collection. And going into these collections, it feels like a library, but instead of books, you have jars of fish. And so each of these jars has fish from a certain location, a certain species, a certain collecting event. And you can then say, okay, I'm really curious to look at the catfishes. I want to compare my catfish to the other catfish of the world. So I can come to a museum like this, I can pull out all the catfish and I can lay them out side by side so I can compare them and I can study the diversity. It's a really important step in describing new species and also documenting the diversity of life in the first place. And one of the things that's also really cool about a collection like this is because we have specimen from 100 years ago, we can compare both present day to past. So we can look in the past what was different from today. So one of the studies that's going on in this lab right now is looking at microplastics and the differences between fishes collected in the 1970s and in the present day. So really exciting stuff. There's just 2 more jars of fish that I want to pull over because they're just so cool. So bear with me. Now, these are also not fish that I collected, but they are really cool and they've been prepared in a special way. You might be able to see they're kind of reddish, and if you get a good look, these are all seahorses. and they have been prepared in a way that's called cleared and staining. So you process the fish to make them semi-transparent, and you can dye the bones and the connective tissue different colors, and it allows you to study the morphology of these fishes without dissecting them to a large degree. So these are really beautiful, cool specimen. These are from the Philippines, interestingly enough, and were collected in 1980. And one last jar, this is another cleared and stained specimen, but it shows a little bit of the other coloration. All right. So this is a sturgeon, and you can see that it has been cleared and stained blue, and it's a beautiful little specimen. Where is this from? This is collected in Turkmenistan. So that's the beauty of these collections. We've got fish from Angola, the Philippines, Turkmenistan, the US, of course, Zambia, Congo. And so these collections, like at the American Museum of Natural History, allow us to see all the diversity of life in one location and really dive into the taxonomy and the diversity. And it's really a cool thing. And I feel very lucky to be able to contribute the specimen that we've collected over the last few years into this collection at the American Museum. And we've got another 4,500 fish en route coming in from Angola. So very exciting stuff. I can't wait to figure out how many new species we have in these collections, and then to get those new species described. So that's a little bit of what I've been up to. And thank you for joining me here in the ichthyology collection. It's really a fun place to be.
All right, Joe, thank you. Thank you for taking us down into the collection with you. I know you have lots of work on the go, so it's great to steal some of your time. And thanks for sharing some of the journey, the experience in the field, and you know what it took to collect those specimens. I can tell from those pictures that it was not easy.
No, it wasn't easy, but it's always fun. You know, even when you're having a hard day, the opportunity to be out in these areas and collecting fish is really extraordinary.
All right, Joe, well, let's take a few questions before we let you go. If you're tuning in via YouTube, don't be shy. Use the chat sidebar. Send us in a question or two. We have a group of students in Missouri hanging out with us in Farmington. So I'm going to bring them in here front and center. There they are. Hey, everyone. How we doing? Perfect. Do we have a couple questions for Joe, either about his expedition in Africa or about what he's up to in the lab right now?
Okay. Lily, you want to ask your question? No? John, you want to ask your question? Let's roll. A little shy this morning.
That's okay.
Hi, my name is John, and I go to St. Joseph's Catholic School. And my question is, what was your closest call with a predator?
Closest call. Well, I did have my canoe hit by a hippopotamus this year, which was kind of scary. But the hippo, it wasn't like trying to attack me. It wasn't trying to be aggressive. We just happened to paddle over it, and then it woke up, and then it came up and hit my boat right underneath where I was sitting. But that was probably the closest call. I mean, I guess you could also say having the lions walking around camp was a close call, but I was sleeping like a rock.
All right. Is that someone else coming up to the camera?
Yeah, if you've got time.
Of course.
Yeah. Introduce yourself.
Hello, my name is Wayner, and I go to St. Joseph's Catholic School. My question is, how did you preserve your food?
How did I preserve my food or my fish?
Food.
I think both would be interesting, Joe, because you're out on the river, so that's hard too.
Yeah. So, I mean, most of the food we eat is dried, mostly rice and beans and dried meats. So we don't have refrigeration out on the when we're in the field. We eat what we can. Whenever we get the chance, we resupply on fresh stuff, bananas and cabbages and things like that. But usually you're eating dried food. In terms of specimen preservation, we preserve all of our fish in formaldehyde, and then the tissue samples are preserved in ethanol, which protects the DNA.
All right. How much of that did you have to carry with you?
How much what?
How much ethanol did you have to carry with you?
I tend to carry a lot. I probably will carry like 2 liters of concentrated formalin and 2 liters of ethanol as well, because I don't want to run out in the field. It's kind of hard to find when you're on the river.
Absolutely. All right. Before we let you go, St. Joseph, do you have any more questions for us?
Anybody want to? Come on, Caden.
All right. We'll squeeze one more in.
One more. Thanks. Yeah. You can do mine. All right, little camera shy. We're working on it. Introduce yourself.
Hi, I'm Caden from St. Joseph Catholic School. My question is, why did you choose Africa and Zambia for your study?
Yeah, that's if I was 10 years old and you could tell me that I could be studying fish in Africa, I would never have believed it. I mean, it just seems like the coolest thing ever, in my opinion. I didn't really choose it. What's interesting, it kind of fell into my life. So I was an undergraduate in California, just doing my normal studies, and I did a study abroad in Ghana. And when I was in Ghana, I just fell in love with it, and I just had such a good time that I said, okay, well, I want to find another opportunity to work in Africa. And I did the Peace Corps in Cameroon. So I lived in Cameroon for 3 years. I started collecting fish there, and the rest is history. Once you get some momentum studying in an area, it's really easy to continue doing that. So, from Ghana to Cameroon to Gabon to Zambia to Angola and, into the DRC later this year. So once you kind of get your foot in the door, you really can take it as far as you want.
All right. Very cool. Thank you, St. Joseph students. Those are some great questions.
All right. Thank you, Joe. And Joe.
Okay. And lots of Joe. We got 2 Joes. We got a St. Joseph school. Some good Joe action today. Okay. Let me grab 1 or 2 from YouTube. Then we'll say goodbye, Joe. So this is from a 6th grade class wondering if talking to fishermen along the way is important to your work.
Definitely. We have one chance. I'm coming down the river. It's my first time ever. I really don't know very much about that system. The people who know about that system are the fishermen, the people who live there, the people who have lived there their whole lives. It's really interesting, not just to discuss about fish, but about the river itself, how the river has been changing. So one of the things last year, one of the rivers, the Kahoot River, was going through an epic drought when we surveyed it. So the local community was informing us about everything that was different this year than what we would normally be seeing. And because we only get one chance to see that river, it was our normal, but it was totally abnormal. So that was really interesting. And then the knowledge of fish, you imagine a fisherman who's been fishing this river his whole life. It is incredible how much they know about the fish, not just the different kinds, but where they live, when they go into different areas, what they feed on. The basics of their ecology and how they fit into the ecosystem are really well understood by the local fishermen, and it's just a wealth of knowledge. I stay in fishing camps as often as possible on these expeditions because I love to learn from the local communities.
Yeah, very cool. 1 more for you, Joe. What kind of tools were you using, your fish catching tools?
Yeah, so mostly I catch most of my fish using a cast net or a dip net. Although unfortunately this last year my cast net was destroyed. We had some nitric acid with us and it spilled all over my cast net and melted the whole thing halfway through that expedition. But no, we've got traps, we've got nets, we've got dip nets, cast nets, and then we've just purchased 2 electrofishers. So this coming year, we're going to be out sampling the rivers with electricity, which is really exciting. I think it's going to allow us to catch a lot more of those rapids adapted catfish. So that's very exciting stuff.
All right. While you were sharing that, I pulled a little clip up on my computer really quickly here. Let me see if I can find it. This is one that you sent us a little while ago using your cast net. So I'm going to share that quickly. I think this is the Gui River.
Yeah, Ugue and Gabon.
I'm Joe Cutler, and I'm exploring the fish diversity of the Ugue River in Gabon. And when I explore, I do it by the seat of my pants.
Boy, I had a big beard at that time.
You've been on the river for a while.
Yeah, so that's the way I catch a lot of these fish. You find a good spot, you throw your net in, and you see what you
Looks
get.
like I've got something.
All right, very cool. And from the sounds of it, you were successful on that cast. So I'm sure there's lots of times where it goes out and nothing comes in.
Yeah, well, that's fishing, right?
Mm-hmm. All right, very cool. Well, Joe, I know it's your last day at the museum, so we're going to let you get back to the work you need to finish. But thank you so much for being with us. And hopefully later in the year when you're in the field, we can get you live.
Sounds great, Joe. Looking forward to doing some live streams. We've got 25 expeditions planned this year, so we're going to be busy.
All right, awesome. Thanks, Joe. Good luck down there, and we'll talk to you soon.
Thanks, Joe. Good to see you. Thanks, everybody.
See you, Joe. All right. Very cool to get to spend some time with Joe. That's, I think, our first time in a lab. Usually we have him on the side of a river or in a tide pool, finding some biodiversity for us. So that was pretty cool to see where some of his specimens end up and just how they're used. You know, it's a library that people will be using for decades, even hundreds of years to come. It's pretty cool. Okay, so we need to do two things before we say goodbye for today. And the first is our experiment. Each week we have a new experiment. You then have 2 weeks to try that experiment in your classroom, or some students choose to try it at home. And then if you send in some pictures or a little video clip trying that experiment, we have prizes for classrooms as well. So we usually pick a few classrooms to send an Amazon gift card to. So this experiment is a really cool one. You don't need a ton of materials, but it's one to use to show how large our periods of time are. So, you know, thinking about our planet and it being somewhere around 4.5 billion years old, it's kind of hard to visualize that. So in this week's experiment, we are going to take a stab at visualizing that space of time. And then it's something that you can try with your classroom as well. So let me get that loaded up, and then we will take a look at that experiment together. All right, where is, here it is. Here we go.
Welcome back, everyone. This week's experiment is one of the most awe-inspiring and mind-blowing experiments you can do at school or at home. It explores the age of our planet Earth and the timing of the key milestones that mark the development of the world that we live in. Scientists know that the universe is about 13.787 billion years old. They know this thanks to 3 separate sources of evidence that give almost exactly the same result. These are the rate at which stars cool, the rate of cosmic background radiation since the Big Bang, and the expansion of the universe. All of these 3 different sources of evidence point to almost exactly the same age, which is 17.787 billion years old. Scientists think that our planet Earth formed about 4.6 billion years ago. This is known from the geology of the Earth and many other sources of evidence. Today's experiment explores the development of our planet and when the key milestones happened that shape the world in which we live. For today's task, you have to measure out a distance of 4.6 meters. You can do this using a measuring tape or even just a piece of string. You can easily measure this using a door. A door is approximately 2 meters long. So 2 and a bit doors will give you your 4.6 meters. This represents the 4.6 billion years of Earth history. every single millimetre along your scale is 1 million years. That equates to 1 metre being a billion years, hence 4.6 metres. I actually haven't quite got enough room here, so I've had to curve my timeline around this table here. This is the start point. This is where Earth formed, at 0 millimetres along the timeline. 1 metre is 1 billion years. 2 metres are 2 billion years. 3 metres are 3 billion years. 4 metres are 4 billion years. And then the last 60 centimetres are the last 600 million years of Earth's history. For a moment, just look down your timeline and remember that every single millimetre equates to 1 million years of history. How old are you? I'm 41 years old. So my 41 years wouldn't even be visible on this timescale. It's literally nothing in this timescale. It's mind-boggling to think of the age of the planet. Your activity this week is to investigate that concept of geological time. I've marked out 8 key milestones in the history of our planet. The formation of planet Earth, well, that's easy. That's at the very start of the timeline. But when did these other milestones take place? These are the first evidence of life. Where do you think that emerges along the timeline? Or the first animals? The first land plants? Or when animals move onto land? Or the first mammals? Or the extinction of the dinosaurs? Or at least for us, perhaps most importantly, when the first humans emerged? Well, your task this week is to take these 8 key milestones and place each one along the length of your timeline at the point in which you think each one transpired. The first one is easy. The formation of the Earth has to be at the start of your timeline. So I'll put this one over here. You might need to do some research this week to find out some facts behind each of these key milestones. But the results are often very interesting and very surprising. Send in photos of your timeline. I'd love to see your thoughts and your ideas when each of these milestones took place. You have a chance to win a £50 Amazon gift voucher prize. Join me next week to find out the answers. Thank you for watching, and I hope you find this experiment really fun and interesting. See you next time.
All right, so there we go. You have 2 weeks to try and create that timeline within your classroom. Like I said, not a whole lot of materials needed for this experiment, a little bit of measurement, make those 8 cards, and then you'll probably have to do a little bit of research to try and figure out where they go. But I think you'll be surprised to see the distances in between some of those events. Yeah, it's pretty amazing to think about the amount of time that has passed. 4.6 billion years. Pretty awesome. So you want to send those pictures or little video clips to classroom at darwin200.com. Classroom at darwin200.com. There, I'll put it up there for you to see it. Send those our way, and then we'll pick some classrooms for those gift cards. So give that a try. You have 2 weeks. So 2 events from now to give that a go. So the last thing we have to do before we wrap up for today is our curiosity of the week. So every week we have a new curiosity, something for you to think about and try and identify what it is. Then send your best answers to us at that same website, classroom at darwin200.com. And then we usually have at least 1 prize for someone who sends in their best guess. We've got all kinds of things from ancient artifacts to parts of living organisms. It's been quite a mixture of our curiosities of the week. And so this one is another interesting one as well. So I have it here on this little piece of plastic platform. So it is shiny. It is relatively more heavy than it looks. So it's a little hard to get it to focus really nice on the webcam. So I've got a little close-up clip. I took a little bonus clip of it today on video so that you guys can see it a little bit closer. So let me bring that up there. There it goes. So here's a nice closer look at this week's curiosity of the week. You have 1 week to think about what it might be and to send in your best guesses. So I don't want to tell you too much about it. You can see that it is shiny. You can see that it is a little bit heavier than you'd expect if you were holding that in your hand. But you have a week to have a think about it. and then send in your best ideas for what this might be to classroom@darwin200.com. So classroom@darwin200.com, send in your best guesses, and then we will announce what this was next week. Okay, so there we go. That is us wrapping up for today. We had a great guest speaker. It was awesome to connect with the ship via video, but I'm excited for maybe a week or two from now when we can make those live again. It's always great to connect live, explore the ship a little bit, and then get a chance to ask Tom and whoever else is on the ship some questions about how things are going. Huge shout out to Joe Cutler for joining us today. Always great to steal a little bit of his time. And yeah, so you have your tasks, 2 weeks to try that experiment, looking at geologic time, and then 1 week to think about this week's curiosity. The last thing we always do is a shout out to our sponsors, supporters, our partners, without whom we wouldn't be sailing around the world. We wouldn't have the world's most exciting classroom. We wouldn't have researchers on board the ship, and we wouldn't be bringing Darwin leaders to the different ports to gain valuable conservation experience. So that is our last thing to do, is a shout out to those who make the Darwin 200 possible. Thanks to our classrooms who joined us live today. Thanks to those who will be tuning in over the course of the week, and we can't wait to see you in next week's live event. Thanks, everyone.